So thank you everyone for coming, and it's very difficult to speak after such excellent speakers you have seen in the past 1.5 days. But that also makes my life easier because I don't have to introduce many concepts and can directly take the privilege of being an organizer and jump into the research talk per se. So what I'll talk about today is concepts in melanoma, and you'll begin to see parallels between EMT and melanoma, some of the ideas that you heard in Herbie's talk, and also, of course, following up from what Yogesh was talking about yesterday. We'll talk about some design principles in the sense that the networks, the gene regulatory networks that we have modeled and other colleagues have been modeling, why are they the way they are? Are there some specific properties which that topology largely has? And then, of course, how we can use that information to then design better therapeutic interventions as well. So this is the picture you saw yesterday. This is a melanoma patient before treatment, 15 weeks after therapy. So you can clearly see the lesions have disappeared, but 23 weeks after therapy, just 8 weeks after this, when again, the disease comes back, right? So this is what is called in the melanoma literature is minimal residual disease, that at this point, when phenotypically there are no lesions, but that doesn't mean that cancer has completely disappeared per se. There were some cells which were lying perhaps dormant or were undetectable, or they did not have the capacity to give rise to such visible lesions in the clinic, but they have the capacity to get reactivated and then grow a tumor yet again. So in terms of melanoma, 70% of all melanomas, they have mutations in this particular pathway, and therefore melanoma has been the poster child for targeted therapy as well as for immunotherapy, and I'll talk about immunotherapy later as well. So targeted therapy basically means that you target a specific molecule rather than chemotherapy, which is relatively more generic, and one of the drugs is vemurafenib. There are others as well, and this is a highly aggressive cancer, because if you look at the survival rate post metastasis, the 5-year survival rate, that's less than 30%. Now, how can we study this phenomena in preclinical settings, right? So this is one example where a BRAF mutant cell line was taken and a BRAF inhibitor was given to these cell lines, and these are two specific markers representative of different phenotypes functionally. So what you see is that pre-treatment also there is some heterogeneity. There are most cells which lie in this first quadrant where NGFR is also high and MART1 is also high. NGFR stands for nerve growth factor receptor, which is a specific neural crest marker, and MART1 is a melanocytic antigen specifically. So this particular state is what is called in the literature as melanocytic state. And as you continue the treatment, you can see that the population structure changes dramatically. You begin to see cells in this particular quadrant, and eventually after 10 weeks, you see that the population structure has changed completely, right? So there are now phenotypes which were not present earlier. So this is clearly a case of a drug-induced cell state transition, because now you're beginning to see phenotypes or cell states which did not exist prior to this. Same observation has been seen in vivo as well. So this is again a BRAF mutant PDX model, where what you notice is that the tumor volume in this initial phase decreases dramatically when you give BRAF inhibition, and then there is this dormant phase for multiple days corresponding to 3 to 4 weeks, again called as minimal residual disease, and after that, even in presence of the drug, you can see that the tumor begins to come up. Okay. Now here people looked at just 2 markers, but of course now in this particular study they did an RNA sequencing, and they looked at multiple markers and gene signatures which had been previously characterized. So what you notice is that in this minimal residual disease period, there is a change in the gene expression profile, and these what are called as invasive profiles have changed, and so have the MITF profile changed in opposite direction. One has gone up and the other has gone down. Now, this idea that there are multiple states in melanoma was first studied almost 15 years ago. What you see here are 4 different cell lines, all are melanoma cell lines, and they are, as you can see, molecularly very distinct, but more importantly, they're functionally distinct. So there are these 2 cell lines which, when studied in vitro, migrate much less as compared to the other 2 cell lines, and the cell lines which migrate less actually grow the fastest. So at a given point of time, cells seem to be doing one thing predominantly, Either they go or they grow. Either they're invasive or they're proliferative. Okay? Lineage I have not seen, but single cell data is there, and I'll present some of that towards the end.
But not, because again, if you look at lineages and you start seeing correlation.
Yes. So of course, Yogesh has done some work in that direction, but beyond that, I don't know.
So what I'm trying to get at is what also Yogesh was saying. Even though the drug is inducing some new states, can you predict these states from states that existed earlier, even before the drug?
So we can predict these states as a, in the example that I'm going to show, as the emergent dynamics of the network itself, that what different states are possible. And then what the drug is doing is that it's changing the steady state frequency. So one state is becoming much more prominent as compared to the other. And then you can look at, do all combinatorial inhibitions and see how can you perturb the landscape in the way that you want. So I'll show you one example towards that. But I have not seen lineage tracing data so far, except Yogesh's work. And Yogesh's work is neat because he starts his colonies with a single cell. So, all the genetic components, at least at the time scale that he does, they can be taken out as a confounding factor, and you can look at phenotypic aspects. Now, when you take these cells and put them in mice, you see that both of them actually grow tumors in mice. The proliferative ones grow much faster, but the invasive ones also, after some period of so-called dormancy, they also resume as if they were proliferative cells. And interestingly, when you look at the tumor sections, both, irrespective of whether you start from invasive cells or you start from proliferative cells, in the tumor, you see both kind of cells eventually in mice. Okay? So which means that this property of transitioning from state A to state B, either in presence of drug or in absence of drug, is seen both in vitro and in vivo, and no state is so-called permanent state per se, right? So both of them can grow tumors, and also you can see heterogeneity here. Now, if you look at the EMT literature and melanoma literature, right, 15 years ago, it was still thought as a very binary phase. So cells are either epithelial or mesenchymal. Cells are either proliferative or invasive. Okay, but of course, in the past decade or so, when people have started looking at more markers, people have started looking at more dynamic data, you begin to see that it is no longer just a binary picture. It's no longer just black and white. There are 50 or more shades of gray in between, which we need to characterize better and understand what exactly is going on. Okay? So here is one such scenario where this experiment was done in the presence of the drug again for almost 3 months. And you can see the cells which began as melanocytic and eventually become undifferentiated or mesenchymal. And they also go through these multiple states. Now, of course, if you look at other papers, the people have been trying to characterize other states. I think the latest paper that came in Nature said there are 7 states, and I'm not surprised if next year there is a paper says which there are 15 states, 20 states, etc. But what exactly is the functional relevance of these states, and are these states really distinct in terms of their ability to grow tumors and so on? No, this is all based on transcriptomic. Okay, so basically it looks like that you can get more markers and you can say this is a new state, call it whatever fancy name you want to call it, and move forward. Okay? So that stamp collection exercise is important. It's necessary but not sufficient to understand how exactly the dynamics of this scenario is changing. Now, of course, this idea of phenotypic plasticity, which plays an important role in drug resistance, the example that I showed you. So you can have some population, and then when you give the drug, many of them die, and then eventually at a different time, you can have a different population distribution as compared to what you had previous to the drug. And that is what the in vitro example that I showed you earlier. Now, what can happen as a consequence of this is that the steady state distribution, or sorry, the phenotypic distribution that you observe pre-treatment can be very different from what you see post-treatment, and also at multiple times post-treatment. Right? So heterogeneity exists at all points, but the existence of heterogeneity doesn't tell you anything about the distribution. You have to look carefully at how many cells are in which phenotype at what given point if you want to study this as a dynamical system. So what we in our lab try to do is understand or develop a sort of intracellular and also cell-cell communication networks at a mechanistic level to understand these cell state transitions that you just heard about from Shaon, and also validate whatever we are seeing in these models using single cell omics data. So we do bioinformatics, and again, going back to the comment that Shaon was saying yesterday, again, we get mislabeled as bioinformaticians often, but we do bioinformatics in the context of getting information about cell-state transitions and not necessarily doing that as an independent exercise. So we try to have a crosstalk between both of these approaches. And the other theme that at least interests us is are there any common principles if you look at phenotypic plasticity networks in multiple cancer? So we have looked at networks in neuroblastoma, where again, there are two states which have been identified, adrenergic and mesenchymal. They can switch back and forth. The rates of cell state transition depend on the dosage and the dosage of the drug. We have looked at in breast cancer, prostate cancer, lung cancer, glioblastoma. And of course, the phenotypic plasticity is not something that cancer invented. It is a property of normal cells as well. So we have been looking at CD4 positive T cell plasticity context. Now, looking at all these diverse biological contexts, of course, requires domain expertise, which is what our collaborators provide who work on each of these specific biological systems, and so that we can identify, are there any common principles that you can save from these networks? So I'm going to talk about the example of melanoma today and then see whether whatever we learned in melanoma, how can, if that is observed in other cancers, and if yes, to what extent. So here is a first attempt to this. And this is work done by Sarthak and Simadri in the group. And they asked a question that, let's say there are 4 states in melanoma. Can I come up with a minimal model, a minimal gene regulatory network, which would explain all those 4 states? So again, simple, go back to the simplest scenarios and see if you can write down. Of course, we know that life is neither smooth nor inclined, as shown as in this example, but nonetheless, we start somewhere. So they came up with this network, and how they came up with these 5 molecules is because they read melanoma papers in detail, identified different molecules reported to drive cells in either proliferative or invasive direction, and identified each of these interactions as reported in the literature. Now, there could, of course, have been many more. We are not saying this is the network. This is a minimal network which can explain, and I'll show you examples of it in a minute. Now, when you take this network and you want to look at the steady states that this network can give you, so what we do is we write down a set of coupled ODEs which represent all these interactions. A typical ODE is written there. So what you get here is that if, let's say, protein X is being regulated by Y and Z, you can write on an equation where the rate of change of X has some basal production rate times the rate, times the what it is being influenced by Y and Z, whether it's an activator or an inhibitor of Z, and then some usual degradation. Okay? Now, of course, it would be ideal if we had the exact values of these parameters, but we do not, unfortunately. So what we do is that we solve these ODEs over a wide range of parameters, and then ask the question, what are the different possible steady states or possible phenotypes that this topology can allow for? And we take all the solutions, we do 10,000 different unique parameter sets, and for every parameter sets, we do multiple initial conditions to make sure that we are capturing multi-stable solutions as well as and when it comes. And then you normalize it across all the solutions obtained. So what you see here is that each row is a unique parameter set that we have simulated. And then each column is, of course, each of these 5 genes. When you look at this in silico heat map, what you see is that there are some patterns. You can do clustering as well to identify that. So there is this set of solutions.
Sorry, Mohit,
Please.
did you say each row was what?
Each row is a virtual cell which has a unique parameter set and initial condition for which it was simulated. So there are these set of solutions where you can notice that MITF and SOX10 are both high and the other 3 are both low. Similarly, there is these set of solutions here where these 3 seem to be high and the other 2 seem to be low, and there are other combinations as well. Then what we did is we know that MITF and SOX10 do, broadly speaking, similar things. So we say, let's define a proliferative score, which is the sum of MITF and SOX10, and an invasive score, which is the sum of these 3 molecules. Now again, when you take this data, this 5-dimensional data, reduce this to 2-dimension, where again, each dot is each row here. What you see is that, as also evident from this graph and the clustering done here, that there is strong antagonism between proliferative score and invasive score. So you can have cells in this entire spectrum. Some are more invasive, less proliferative, some are more proliferative, less invasive, and of course, the 51 shades of gray in between as well here, right? So what are those shades of gray here? So we characterize them as neural crest-like because NGFR, we looked at other molecules in the data as well, and these are two distinct states, as you can also notice here. Okay, so this is just a prediction. Okay, let's look at real data. So this is, then we go to Cancer Cell Line Encyclopedia and take all the skin cancer lines, and from the 20,000 plus gene expression matrix, we only pull up 5 genes, these 5 genes, and see whether that antagonism is seen here. And the answer is yes. Let's look at TCGA, skin cancer, only again pull up those 5 genes. Again, you see a strong antagonism being noted here, which is what our model had predicted. Okay, let's look at single cell RNA-seq data. So these are 10 different cell lines for which single cell RNA-seq was done. And again, you only take these 5 genes and you see a strong antagonistic relationship, which is what our model had predicted. Now, if only 5 dimensions can explain this strong antagonism, whereas on the other hand, people have been identifying so-called gene expression signatures or differentially expressed genes, is there any connection there? So what we did is we looked at such gold standard invasive scores and proliferative scores, which are like 500 or 600 genes taken together, and in this exact data set, and we notice that whatever we had predicted, that this particular phenotype where these 3 are high, so 1, 1, 1 and 0, 0, the first 2 are low, that actually has the highest invasive score. And similarly, the exact opposite, where in this particular state, when these 3, SOX9, JUN, and ZEB1 are high, which means you're lying somewhere in this region of the plot, you see that the 600 gene signature also shows it's low. Okay? So whatever information is contained in 5 dimensions can be actually validated in some gold standard, much higher dimensional space as well. No, it's MITF and its targets, but it has almost 500 plus genes. Both of them have 500 plus genes.
Proliferative score means if it's high, then these cells are
more proliferative. Yeah.
faster or?
So they grow faster. Yes. So in the in vitro equivalent of that, this plot that I showed earlier. Yeah. So they grow faster.
For this, you have to have 500 genes.
Again, it's not causative connection. All they have done is these samples grow faster, these samples invade faster. Let's do differential expression gene, set some arbitrary cutoff, and proceed further. There's no causative connection.
There's no one who's found a less number of genes required to create similar scores.
Not that we have seen so far. Yes.
Did you say that you were?
Yes. So no one else has. I mean, we did it from bottom-up approach. We didn't keep changing the threshold of differential gene expression and decide accordingly. We came up with a mechanistic model which could explain. Yes.
Slightly mathematical question.
Please.
So I see in the next slide that both June and SOX9 contribute towards invasiveness.
Yes.
So can you not replace those 2 nodes as a single node and have a 3-node network instead?
I'll come to that. Yeah. So that's a good point. Can you combine nodes or have a super node kind of thing? I'll come to that. Yes. Yes. These are based on the literature that in the people have identified these extra states as well, right? These hybrid states. Or neural crest has been identified, intermediate has been identified. So we looked at the levels of these 5 molecules in these phenotypes that had been identified, and based on that corresponded them to. Okay. So now let's do perturbations in the model. Let's overexpress, downexpress. Let's do in silico perturbations. So let's down-regulate SOX10. So when you down-regulate SOX10, we know which direction it lies. So mathematically, what we do is we change the range of parameters corresponding to SOX10 production. So you decrease it by an order of magnitude. Okay. So what you see is that now there are many more solutions which correspond to an invasive phenotype as compared to proliferative phenotype. So what SOX10 down regulation is effectively doing is driving a proliferative to invasive transition. Okay. Now let's look at the real data. So again, these are all transcriptomic data sets. where exactly this was done. SOX10 was knocked down, and you see that just looking at these 5 genes, proliferative score went down, invasive score went up. And the same thing was seen in a different data set. So it's not just specific to a given set of experiments. Okay. Let's do the other experiment in silico, overexpress SOX9. And that also does the same thing because SOX10 and SOX9 lie on opposite sides. So SOX9 also drives the same transition. And again, when you look at transcriptomic data, you see the same behavior. When you overexpress SOX9, proliferative score goes down, invasive score goes up. Now, this is bulk data. Let's look at single cell data. So this is interesting because this is single cell and time course. So what you see is that here are the control cells. And again, we are keeping the same y-axis, just the 5 genes. The control cells are much more proliferative. And as you knock down SOX10 and keep moving further in time, you begin to see this trajectory that cells are moving, and eventually they become much more invasive as compared to what they were previously, right? So what this tells us is that, again, in a simple 5-gene network, you can do these perturbations, and that 5-gene network seems to be sufficient to explain multiple different experiments done in vitro, in vivo scenarios, because you can't do these perturbations in patients. You can only do this in the preclinical settings. The next question we asked is, again, because immunotherapy is an important part of treatment of melanoma, and going back to the question that Rajesh was asking yesterday, is there a connection between EMT and immunotherapy, right? Is every cell equally likely to die by immunotherapy? And the answer is no. People have shown this in the context of EMT and also in the context of melanoma, that the cells which are more invasive have increased the levels of checkpoint inhibitors on their surface, and therefore decreasing the ability to be sensed by immune cells and be killed by immunotherapy. Okay, so the fact that the cells begin to move has other implications as well in terms of its drug sensitivity to various different therapies. So we added PDL1 into our network based on whatever was known in the literature. And again, what we notice is that consistent with the preclinical and clinical data, PDL1 is actually high in invasive cells as compared to proliferative cells. Now again, we can go back to single cell RNA-seq data, the same data set that we looked at earlier, and you can see that the higher the invasive score, the higher the PDL1 levels. You can also look at these kinds of experiments where people generated sublines by continuously treating cells with a given therapy, and you can notice that whenever cells move from sensitive to resistant phenotype in the BRAF inhibitor, they are also gaining PDL1 high. So what seems to be happening here is that this is what is cross resistance. You are gaining resistance to, or the cell state transition that the drug is inducing is of course in rendering that therapy ineffective, but simultaneously it is also making cells resistant to immunotherapy. And why is that happening? Because the molecules are connected in a specific manner. If they were not connected in this manner, you would not have seen this, right? So this is it's a direct outcome of how these molecules connect to each other. Then again, something that Yogesh talked about, if you only have bulk data, for instance, right? So let's say you have some data pre-treatment and then post-treatment, and you see that the phenotype changes. How can you say that whether it was selection, that these cells existed prior to treatment, or this was induction? And you cannot say this in bulk data alone, right? You would need single cell data, or you would need lineage tracing, barcoding, etc. to do this. So what we have looked at here is this particular cell line, where if you look at the pre-treatment cells, the control cells, and here the axis we are looking at is the much higher dimensional scores, because in single cell data, just looking at 5 genes sometimes may not be sufficient. So what you see is that even prior to treatment, you see 2 subpopulations. There's this subpopulation here, which is more proliferative, less invasive, and then there's other peak here, which is more invasive, less proliferative. But when you look at the same post-treatment, this population disappears. You only have the other population, right? So in this particular scenario, what you see is that pre-existing heterogeneity was there. You already had 2 subpopulations, one of which was killed and the other survived. But let's look at the same data set more carefully when you start to look at combination therapy. So I only presented half the data in this, the other half is here. So this is the same, this blob is the same as this entire blob. Now, when you look at combination of therapy data, you begin to see new phenotypes which did not exist previously. So this combination seems to be capable of pushing cells to a different state which was not there. So both selection and plasticity can play important roles. It depends on which therapy we are looking at, what concentration, what the genetic background of the cells is, etc., etc. But we see evidence of both of them depending on which drug that we look at. So that was the one approach, starting with minimal network and see if you can explain whatever was seen. Here is another approach which Malvika, my ex-student, she was IISc undergrad, did, was she wanted to start from omics data and then arrive at a network and see whether that network was sufficient to do that. So what she did is this: she started from two sets of samples, proliferative and invasive, the omics data of that, and applied a whole bunch of statistical approaches which many of you might be familiar with, where you identify differentially expressed genes and what the modules are, and then you look at what are the transcription factors controlling those genes, how do those transcription factors influence each other in the network, and so on. And she came up with a 17 gene network and did the same approach, which is solve that 17-dimensional system over multiple parameter ranges, and saw that this was her in silico heat map that she generated for the 17-dimensional case. Again, interestingly, you see this clear pattern that there are some cells which have, again, you can begin to see MITF here and JUN. MITF and JUN lie on opposite sides here as well. And these are the only two common genes in that, this 5-gene network and this 17-gene network, okay? That 5 is not a proper subset of this 17 because these two are completely independent approaches taken by different groups, different people in the group. Okay, so you have this 17-gene network. Again, you can project them on PCA. You identify that, again, there are 4 phenotypes. If you look more carefully, broadly speaking, there are 2, but then there is a substate that you can see here and another substate that you can see here. And what her interpretation of the data was that, when people were saying there are 4 states versus when people were saying there are 2 states, none of them were wrong. It's just about the resolution at which you look. So if you look at the broadest resolution, you say there are 2 phenotypes, but when you go a little bit deeper, you can say that there are 2 substates of proliferative, or there are 2 substates of invasive. And then of course, you can go further deeper and say there are substates or microstates or nanostates or whatever, right? So it depends on how many markers that you are looking at per se. Okay, then we again did these kinds of experiments where we knocked down MITF, which is what BRAF inhibition does, and we see that the population structure changes, and this change exactly happens as was shown in vitro in the earlier data. So again, even the 17-dimensional network was able to recapitulate some of the experimental data. She went a step further and said, I'm going to now do combinatorial perturbations. I'm going to do all 17 C2 scenarios. And see, is there a combination or many combinations which can actually get rid of some other states and push all the cells into one state so that heterogeneity can be brought down and the therapy can be more effective. So she found one such example where if you knock down SMAD3 and MITF, you see that there is only one phenotype that remains. All the other states have disappeared, okay? Because the network structure is such that you can get rid of other states. Now, there is some preliminary validation of this in the existing literature, but yeah, we haven't done 17 C2 combinations experimentally and showed that this is the best one. Yes, Shaon.
So maybe I missed this. So in your dynamical models, how do you introduce the drug in the model?
So you reduce the production rate of the target. So by an order of magnitude, or 2 orders of magnitude. So far I've presented 2 stories. A 5-node network bottom up, okay, start from reading the multiple papers, real intelligence, and the other is artificial intelligence, where you apply all these statistical approaches and then come to this. But why are we getting such common results in both of them, that both of them seem to be able to explain multiple observations? So there are 2 hypotheses that we have. One is that these 5 are like so-called master regulators, which somehow occupy a higher pedestal in the network structure, and these remaining 12 here are mere supporters. Okay? And the other hypothesis can be that there are some other structures which these 5 and 17 dimensional networks collapses to, and there is striking similarity between what you see in the reduced dimensionality space of 5 gene and 17 gene. These are the two hypotheses we have. We'll be happy to take any other suggestions as well. Now, one idea that we actually saw in this particular case was that when you look at all the solutions of this model and then do a pairwise correlation between all the 17 dimensions, you see a clear pattern that these sets of genes are positively correlated with each other. These sets of genes are positively correlated with each other, and across these there is a strongly negative correlation. Now, we wanted to see whether this was just an artifact of the model or was this also seen in the data. So we looked at cell line data, the same dimensions, the same variables, and you again see a very similar pattern. And now when we come to this 5-dimensional network, we notice we did this analysis where we looked at multiple different data sets. We just didn't want to restrict ourselves to one transcriptomic data set. So we looked at multiple different data sets and asked, is this antagonism that was noticed earlier seen in multiple data sets? And the answer is yes. And again, whatever antagonism is seen between these two dimensions, where Z and MITF is one axis and the Jun, SOX10 and these are other axes, sorry, MITF and SOX10 and the Jun, Z1 and that antagonism could be seen in 5 dimensions only, and that same antagonism was seen in 1200 dimension space, where you had 600 genes of one and 600 genes of the other. Right? So again, that pattern can be seen in a much reduced dimensional space. You really do not need 500 genes to characterize a given phenotype per se. Okay. So this is what we call the phenomena of teams, that there are teams of players rather than one master regulator driving something. There seems like that there is a whole bunch of players driving a phenotype, and there's a whole bunch of another players driving the other phenotype, and they just cannot coexist. And because of that, I'll show you data in a minute, is that because of that such strong mutual activation within a team and mutual antagonism across teams, you see that the dimensionality comes down. And this is what goes back to the question that was asked, can you reduce 2 nodes to 1? And the answer is yes, you can reduce 10 nodes to 1. Okay? And I'll show examples in a minute. Okay, so we said, fine, melanoma, maybe this is just a fluke. Let's look at other cancers. Does this work? So we looked at small cell lung cancer, which is also exceptionally aggressive cancer, less than 10% 5-year survival rate. No targeted therapy still available for this cancer. So we look at cell lines and we look at, again, the genes, so-called differentially expressed genes. You see again a clear pattern. One set of genes positively correlated, the other set of genes negatively correlated. When you project them on a principal component analysis and look at what exactly is PC1, we often just look at PCA plot and say, well, they are segregated. That's the bare minimum information that a PCA plot can give you. You can also look at what exactly is the composition of principal component 1. You can look at the coefficients of the variables that constitute that, because what is principal component 1? At the end of the day, it's a direction. It's a linear combination of those different variables in a direction in some space, which is explaining the maximum data. Right? So what exactly is that direction is something that we can look at. So when you look at that, you see that every variable or every gene that supports a so-called neuroendocrine phenotype. So again, there are 2 different phenotypes in this. Don't worry about the names. You can see that everyone has a negative contribution, and everyone that supports the other phenotype has a positive contribution. So again, they segregate very well in the higher dimensional space. You can go back to EMT and you see the same pattern again. And interestingly, you can notice that principal component 1 is explaining a reasonably high proportion of the data, 50 to 55%. And I'll show examples where it does even better than that. Okay? So it looks like that this idea of teams of nodes or teams of molecules is seen in multiple cancers. Now this is the small cell lung cancer that I was telling you about. And here is a network of this. Is 33 node network. So we didn't simulate it using ODEs because this is a much larger network. We simulated it using a simple Boolean model where we just take every gene is either on and off. And because we do not know the rules for every gene, we take a very simple majority rule that if on a given node you have more activations than inhibitions, you turn the gene on. If there are more inhibitions than activations, you turn the gene off. And if they're equal, you don't update the node. Very simple update, okay? You can say this is, well, biologically perhaps not reasonable. I agree with that criticism, but this is our first approach that we see. When you do that, and we actually saw that this particular network, I'll just show that data here, this particular network, which could have had 2 to the power 33 solutions, has only 4 solutions, from 2 to the power 33 to 2 to the power 2. Okay? And when you take this network and you shuffle the edges between it, so create random, non-biological, hypothetical networks, what is the distribution of number of states? The mean lies around 10 to the power 5, which is what we had expected earlier, that with such a high dimensional space, you'll get many, many, many solutions. Okay? And this we show later that this happens because of the presence of teams. If you do not have teams, you will not see such huge dimension reduction from 2 to the power 33 to 2 to the power 2.
These modules sufficient to give you canalization?
So, I mean, that's a great question. And we have been talking to different people who use the word modules and modularity, and they don't agree on each other's definition. So we are seeing whether teams agrees with a definition of modularity. And so teams can clearly canalize. If you mean by canalization the reduction of dimensionality space or phenotypic space, that teams can do for sure. They can also offer robustness to small perturbations. Now, whether this is what people called modules earlier in the graph theory literature is something we are still figuring out.
It's still possible that a particular definition of module
may match with this. Yes, may match. Yes. And again, when we look at this network, you simulate it just as the data I showed you in melanoma. You see this clearly team's behavior in the ODE model solutions as well, and the same pattern was seen in cell line data. Now, so in this part, what we have talked about is that multiple regulatory networks, whether it's a 5-gene network or a 17-gene network, they are both multi-stable in nature. They can give rise to multiple states, and it looks like that there is a hallmark of these networks that they look very complex, but they can actually be reduced to something very simple, which is the presence of teams of nodes in this particular case. Then the question comes, why do teams exist? So again, following up on the discussion I was having with Shaon, one is to perhaps shape the landscape. If you have multiple states available, please. Yeah, I'll come to that. There is a strict definition we are arriving at so that we can avoid all this confusion about the definition with modules. So what is? Absolutely. Right. So those are questions we are still battling with. A, we don't think every gene necessarily needs to form a team. There can be, independent contestants in an election which are not supported by any political party, kind of situation, right? And even within a team, just like as in life, when you do group projects in class, does everyone does equal job? Never, right? So there can be members of a team which contribute more and members which contribute less. So then, these are my last 3 slides, so I'll get to that later. Fine. So, but those are great questions. I mean, we have not yet solved the entire puzzle. We are just seeing some patterns here. So this is one idea we have, that perhaps the presence of teams controls the rate at which cells will transition from state A to state B and so on, because I'll show an example that not every phenotype is necessarily supported by a team. There are phenotypes which are not supported by a team, and those are the ones which are most plastic. Those are the ones which, whenever a little perturbation comes, they just leave their phenotype and go somewhere else. But the ones supported by a team have that strong support system and resist being pulled out of that attractor as much as they can. So that's one. The other idea which we have been gaining some interesting data is that, if again you think of the journey of a metastatic cell, it is a highly, highly challenging journey. It has to overcome so many different bottlenecks. It has to change its metabolism, it has to change its property to evade the immune system, to evade the drugs, to change adhesion, migration, to settle at a different site, etc., etc., etc. Right? So perhaps the presence of teams can, again, tie these properties, that when you undergo EMT, you already gain drug resistance because of the consequence of the way the molecules are connected to each other. And that might be a way to increase the fitness of metastasizing cells. Now, this is the actual network where we looked at teams much more carefully. Now, here is a 22-node network, and you take this network and you just represent this as a matrix. Each row is the source, each column is the target. Wherever there is an inhibition, there is a blue box. Wherever there is an activation, there is a red box. So in this 22-node network, you have 22 cross 22 possible interactions, and only 82 edges are there. So this is approximately 20%. And that's what you see in this matrix. It's a sparse matrix, right? Not everyone interacts with everyone directly. Okay? But again, as we know, both in life and in networks, you need not interact with anyone directly to be influenced by that individual or by that node. Your impact can travel through WhatsApp or Facebook or whatever to impact the other person. It may be weaker than a direct interaction, but it is not zero. Okay? So what we did is we just multiplied this matrix many times to itself, because that is how you capture the indirect interaction. If A influences B and B influences C, then when you multiply this matrix, you will get A influence on C, right? And when we do that, you see a very clear pattern that emerges. Now in this network, because of extensive information available in the literature on EMT, we know which genes are epithelial and which genes are mesenchymal. And what you see here is that every epithelial node activates every epithelial node. Every mesenchymal node activates every mesenchymal node. Every epithelial node inhibits every mesenchymal node. And every mesenchymal node inhibits every epithelial node. It's a very simple structure. The extent of activation and inhibition is different, no doubts about it, but the pattern is maintained. We looked at 5 other networks published by 5 different groups to avoid any biases that we were selecting networks which were published by the same set of individuals. And none of these networks were built by the second approach, which is the statistical approach. All of these networks are built bottom up as people keep reading the literature and keep expanding the networks. Right? So we saw the same pattern in all of them specifically. So again, we asked this question. Now let's look at 1,000 random non-biological hypothetical networks of the same size and same density. Do they also form teams? The answer is no. This is the distribution, the histogram of those, you can quantify group strength based on just summing up these 4 values and taking a mean of them. And this is where the wild type lies. On a scale of 0 to 1, you are, 98th percentile or higher as compared to the random networks. Okay?
Sorry, so is that purely randomized network?
Yes, all what we do is just shuffle these ones.
This is something I was thinking when you're speaking, right? So we know that biological networks are not random. So even if you take normal cells, you know there will be
Absolutely.
and so on.
Absolutely.
So if you want to, what is your statement exactly? That in cancer cells only you see these?
No, that only I'm not claiming. In cancer cells, we see this. Whether this is seen in development or not, we are still looking at it. We have one example where we see that this is seen in development as well. So this seems to be a general property which is capable of reducing the dimensionality of the system per se.
The randomization you're doing in the next slide,
Yes.
it may not be very biologically relevant because we know that
Yes.
in biology.
Yes, yes. So it's a negative control. We are just showing that this presence of teams is specific to biological network. Then you can ask the other question is that now let's say you start from an epithelial state, mesenchymal state, and a hybrid state. Okay? And the question we are asking is, what we have done here is that you start from these states and then you flip one node. If it was on, you turn it off. If it was off, you turn it on. Okay? And then on the x-axis is how many nodes as a fraction of the total nodes available have you flipped. And of course, there are many possibilities of this. So we did a mean of a large number of simulations here. And the question we are asking is, where is the threshold? How many nodes on average would you need to flip if you want to push an epithelial cell out of its epithelial behavior? Okay? That answer is 50% if you start from epithelial, if you start from mesenchymal. What about the hybrid states? No matter which hybrid state you talk about, there are many, many hybrid states, right? All happy families are happy in the same way. All unhappy families are unhappy in their different ways, right? So hybrid state, which do not have any team, they are some random combinations of some epithelial on, some mesenchymal on. All epithelial states are all epithelial on, all mesenchymal off, and vice versa, right? No matter which hybrid state you look at, you can push them out of that state with changing 5% of the nodes. 50% versus 5%, an order of magnitude difference, because hybrid states have no teams. So that's a great question. So, we talk about stability and instability, but when we are talking about multi-stable systems, it matters relative stability, right? So how much, and that is what we are trying to quantify here. So hybrid state is also stable, but it is stable to a less extent. Yes. Yes. Depending on what perturbation has been given, yes. So it's basically the absence of teams which makes these hybrid cells the fittest for metastasis. These are the most adaptable cells, as Herbie was talking about yesterday. And our argument is that the reason why they are most metastatic or most fit is because they have no teams. If they had teams, then they would be lying in a deep attractor and very difficult to get out of it, per se. So the question we were asking here is the network topology sufficient to explain these whole bunch of in vitro and in vivo observations that hybrid cells are more plastic? And the answer seems to be yes. Okay? We don't need to look at any parameters. This was just done by Boolean models. Now we start looking at one other case study. So this was resistance to tamoxifen, which was actually the first targeted therapy that came up. And it is given to estrogen receptor positive breast cancer patients, which is the most has the maximum of breast cancer cases. Again, the idea of EMT, Herbie already talked about earlier, so I'm not going into the details. EMT usually happens when cells begin to get out of the tumor. MET usually happens when cells are trying to settle at the tumor. So the questions we were asking is that, are EMT and drug resistance connected? Why do you see that when a patient gets metastasis, the same drug which worked for a primary tumor stops working for metastasis? If they're back to being epithelial, that drug should work, right? But something has changed in that entire process, okay? So, which again, something we are working on, I'm not going into the details of that. The question we are asking is, when cells come back, presumably, do they come back to the same epithelial state? Or do they come back to an altered epithelial state, right? When you go through a trauma, you may recover from that trauma, but are you the same person after that? You have gained or lost some of your attributes, right? In that sense, per se. So again, what we did is identified a minimal network, simulated it, generated this so-called in silico heat map, and showed that you can actually see 3 peaks in terms of EMT, 2 peaks in terms of resistance. So this is the molecule which is targeted by tamoxifen. This is a splice variant of it, which is not targeted by tamoxifen, and they control each other in different manners. This is all built up based on reading the literature very, very carefully and trying to identify only those links for which there is a detailed causative connection established. That when we say A inhibits B, it's not just you perturb A and B goes up. Does A go and bind to B? When you mutate the binding site, is that effect gone down? So there are different levels of credibility of each of these links, but we try to keep that level of credibility as high as possible. Okay? So what we see here is that when you project, again, 5-dimensional network, 2-dimensional plane, EMT score, resistance score, strong correlation. Epithelial cells mostly sensitive, mesenchymal cells mostly resistant, and hybrid cells also seem to be split in both resistant and sensitive. Then let's look at transcriptomic data and the perturbations we made in our model. So what our model suggested is that when you induce EMT, you push the cells to a mesenchymal resistant state. Now let's look at transcriptomic data where EMT was induced in all different ways. These are all ER positive breast cancer cell lines. There are 4 or 5 different ways of inducing EMT in them. Irrespective of how you induce it, you see that EMT goes up, estrogen response comes down. Let's look at the other way around. Can drug resistance induce EMT? So you're treating a patient with the drug, continuously increasing pressure of the drug is going to select for some cells which are not dying. Whether the selection is at a genetic or non-genetic level is a separate question. Are those cells more mesenchymal to begin with? Okay, the answer is yes. You look at when estrogen response was silenced, EMT went up. You look at the sublines which were generated by exactly what I just described in a preclinical in vitro model, EMT went up. So EMT is driving drug resistance and drug resistance is driving EMT. That is why you see both properties simultaneously. Yes, so that is what we are still working on, which perturbation is going to be most effective. Yes, 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 yes. So it's like, absolutely, absolutely. We have so far not looked into how is TGF-beta induced EMT different from EGF, different from hypoxia induced EMT. We have not looked at those differences. This is a very first pass model that we have built in that sense. Okay, now can teams connect three properties? We have seen EMT and tamoxifen resistance. Let's look at the so-called stemness property, which is the ability of a tumor to give rise to multiple different cell types. So we take the EMT network, tamoxifen network, and add a stemness network that we had looked at earlier. Again, clear teams, epithelials, tamoxifen resistance, stemness properties, all players support each other. Mesenchymal, tamoxifen resistant, stem-like properties support each other. Okay, and again, you can do this randomization and show that this property of teamingness or team behavior is again seen in wild-type network, not in random networks. Let's look at another combination. EMT, tamoxifen resistance, immunotherapy resistance. Okay, you can pick any combination. We built an updated network. What we saw was that as this is transcriptomic data, when you silence estrogen receptor, of course EMT goes up, that we have seen earlier. PDL1 also goes up. So you are inducing resistance to tamoxifen, but you are buy one get one free, getting resistance to the other therapy as well simultaneously. Why? Because the molecules connect to each other in a specific manner. If you randomize this network, you don't get these properties. Now, then we started thinking, is teams a meaningful way to reduce dimensionality? And there are multiple dimensionality reduction methods that the colleagues in the field have been working on. Now, can teams provide a meaning to that, a biologically feasible meaning, rather than doing a feature selection kind of approaches in an unbiased manner? Okay? So what we did is we looked at this same network that I told you earlier about and simulated this using ODEs and projected them on principal component 1. 95% of the variance can be explained by principal component 1. Okay? When you do this for random networks, that distribution is, the mean is around 30 to 40%. Let's project these things together. X-axis is team strength, Y-axis is principal component 1 variance. This is where the wild type lies, far away from 1,000 random networks. And you can clearly see this trend here. The higher the team strength, the higher the principal component variance that you're going to get. And again, to calculate team strength, you don't need to do any simulations. All you need is the network, just multiply the matrix, you get the teams. So you can get information about the dynamics of the network without simulating the network by using this particular idea. Let's look at the components of principal component 1. All mesenchymal positive, all epithelial negative. Very clear distinction. Let's look at a random network, it's all distributed. There's no pattern. Okay. So basically EMT networks can be explained, and also the transcriptomic data that I showed you earlier can be explained mostly just by principal component 1. Going back to Shaon's question, is this specific to cancer? No. You look at gonadal cell fate determination, you look at pluripotency in embryonic stem cell, same patterns. These are the networks, these are the influence matrices obtained after multiplying the matrices. You look at principal component 1, phenotype 1, all positive, phenotype 2, all negative. You can change the context, you can change the name of the players, as long as the team's behavior is there, you'll see this pattern. Same thing, wild type network lies extremely far away from others, and all of them fit a Michaelis-Menten behavior. Why, how, we don't know. That is what we are still trying to figure out. Then we said, let's generate artificial networks. These were all biological networks, okay? Let's assume there's a 10 node network which comprises of 2 teams of 5 nodes each. And I'm going to change the density the way I want, okay? So in this case, 25% density, which means that, so what you cannot do is, change what is not allowed is cheating, okay, basically. Absence is okay, infidelity is not, okay? Now, when you do that, you generate different densities, you simulate these networks, see what we get. The question we were asking is, what is the minimum density you need to get a team strength? Even at 30% density, you are at team strength, 0.6, 0.7, which is very comparable to the values that we have been seeing, right? So you don't really need everyone to be present and raising slogans for you. As long as 30% people are doing it and there's no infidelity, you're good. Okay? Let's the same question about PC1 variance. Again, by 30%, PC1 variance is 100, almost. Okay? Team strength versus PC1. So again, all of them fit some sigmoidal behavior, and we are still trying to understand why sigmoidal, what is the meaning of these thresholds, what is the meaning of this nonlinearity, etc.
Principal component is trying to reduce to a space which is probably your team, right?
Absolutely,
that direction
absolutely.
is your team.
Absolutely.
So this is not too surprising.
Absolutely. But we wanted to get this value.
Right.
Yes, that was surprising. The 30% is sufficient.
Right. But when you have many teams, if this still happens, I would be
then this will not happen, and there's a reason. And there's a reason for that, which I'll come to in a minute. That this behavior, whatever we are seeing, is specific to 2 team networks. The moment you add a 3rd team, this will fail. Okay, let me be clear. Then you will need 2 principal components to explain the data. One will not be sufficient. PC1 plus PC2 will still explain maximum data.
But is that too surprising? I mean, I'm not too surprised.
No, I'm not surprised. This was just a validation that if you now play God and construct artificial networks, how much effort would you need to put in to get that? Yeah. So this was just a validation. There's no surprising aspects. Now, this idea of teams need not be restricted to intracellular networks. You can think about cell-cell communication networks, and there also you might see teams. And this is something I worked on during my postdoc, that you have epithelial cells, you have mesenchymal cells, you have M1 macrophages, you have M2 macrophages. They're all secreting different kinds of things which is capable of changing each other's behavior. So what is known in the literature is that if you have M2 macrophages, they secrete something which can induce EMT. If you have M1 macrophages, they secrete something which can induce MET. Mesenchymal cells, for reasons I don't know, secrete something which can push M0 monocyte to M2. Epithelial cells, for again reasons I do not know, secrete something which pushes M0 monocytes to M1. It's a team behavior, right? So that is why what you see when you simulate this model, which we did 5 years ago, but we didn't have this concept of teams at that point, is that epithelial cells and M1 macrophages come together, mesenchymal and M2 macrophages come together, right? And now, of course, you can. This is just a two members in a team case. You can have many more. You can have, you can add whatever cancer-associated fibroblasts and non-cancer associated, whatever, normal fibroblasts in this case. Do you see that segregation? Why? Because at the end of the day, we are thinking about the sign of interaction and the strength of interaction, right? Whether it's through a cytokine, whether it's through gene regulation, doesn't really matter, as long as it is happening and it is, in a self-consistent manner. So of course, this we wrote up together with our colleagues, thinking about group behavior in cancer and how that we can connect to teams idea. So I'll just summarize this second and last part of the talk, is that what we think is that teams can coordinate axes of plasticity. So the existence of teams might not be surprising, but the implications of that are crucial for how cancer cells behave and what increased fitness they might have, right? So the reason you're seeing cross resistance is because of the presence of teams, because of the presence of these feedback loops in a specific manner. Teams can explain why cancer cells that do not get killed become stronger, right? Because if the cells which are not killed by tamoxifen, you're making them more EMT, you're pushing them to metastasize, that's a side effect of the drug. We don't have numbers on how many cells get killed versus how many become more metastatic. How does that depend on duration? How does that depend on concentration? We don't have any of those numbers, but all we are saying is that whatever we have done so far shows the possibility of that happening, and that is the implications of that are not small to be ignored. And of course, teams can be a biologically meaningful dimension reduction. Right? So what you might think is that, well, I have targeted therapy, it is going to hit a molecule, and everything is just going to be sorted. Okay, but there are cells that survive because there are teams, there are people who support each other, and they get out of stressful situations like that, and they become stronger than what they were earlier. So at this point, what we are thinking is that teams can be a possible way in which, despite huge dimensionality in the interaction space, and this is going back to Shaon's point again, this is not at all specific to cancer. Why we see only 200 cell types despite so many interactions happening? This is our current idea, is that teams might be reducing frustration in networks, or pushing cells only to a specific allowed set of states, which is what corresponds to specific phenotypes. So I'll just summarize this. In the first half, we talked about design principles of networks, which actually drive cell state transitions. We looked at multiple different cancer types and saw that multi-stability showed up in all of those places. And so did the presence of teams, which have the capability to reduce the dimensionality space and, more importantly, increase cancer adaptation. More importantly for cancer, reduced dimensionality can be seen both in development and cancer. Therefore, this can be a biologically meaningful variable to look at this. Now, what are the open questions? One which Shaon already asked. What if there are more than 2 teams? Right? What if all these are competing for the Iron Throne? And they may not form very stable teams. So what happens if there are 3 teams, 4 teams, 5 teams? Right? And this is a question which these 3 undergrads from Vaibhav, Kushal, and Aditya are trying to address currently in the lab. Do teams offer robustness against fluctuations? Right? So you take a network, it will have all different kinds of noise. If you have teams, do you reduce the sensitivity of that network to internal noise, to external noise? And this is a question which Lakshmi and Abhay, undergrads, and Kishore, my ex-PhD student, now a postdoc with Herbie, is trying to address. And what about master regulators, right? So there are master regulators, P53, MYC, etc. And when we say teams, I'm not saying that teams and master regulators concepts are incompatible. Because even within a team, you can have players which do more important job as compared to others. But then how do these aspects sort of, again, fit into each other? And this is, again, a question which Pradyumna, Saundarya, and Sarthak are looking at in different contexts, specific examples as well as more general ideas as well. So with that, I would like to acknowledge my team, which has members and everyone works hard in this team. And they come from diverse backgrounds. They all interact with each other. And of course, our team of collaborators who also comes from phenotypically heterogeneous backgrounds and work together. And I'd like to thank the funding agencies. And if you want to join our team, there are positions, both PhD positions as well as other positions. Please contact me. And thank you once again for your attention. So far, one team.
Getting out of hybrid states, meaning you will either go to epithelial or mesenchymal state. But when we talk about metastasis, we primarily focus on the existence of hybrid states and its properties to navigate through the whole process. So given any perturbation, the current structure kicks hybrid states either into the epithelial or mesenchymal. So is it possible to remain in hybrid state through the net? So can there be a network structure that provides such kind of transition between the hybrid states as well?
Right. So that's a great question. So far the networks we have looked at support that epithelial and mesenchymal ones are the most dominant phenotypes, and epithelial ones are hybrid ones are A, less frequent and B, less robust as well. But we have at least one network which actually gives hybrid as the most dominant one. We are still trying to understand how that network is fundamentally different from other networks. Having said that, there's a clinical observation in, I think it's a 2 years ago paper, which the question they asked was, broadly speaking, what is the minimal percentage of hybrid cells in a patient sample that would reduce survival dramatically? Of course, in clinical data, you can only associate, you can't establish causality, and that answer was 2%. So even if you have 2% hybrid states, you see a dramatic change in the Kaplan-Meier curve. And all the networks we have simulated now give at least 2% simulation. So even these so-called wild-type networks have that potency. Now, what networks? I think going back to one of the questions which was asked, now if you begin adding more players which do not fall in any team, right? If you reduce the effectiveness of teams by adding cheaters or by adding non-aligned people in there, you can get hybrid states as more dominant. That's not surprising at all.
I want to ask, how do like different players in a particular cancer form a particular team? And as you also talked about master regulator, I thought about it being like a star player, like say somebody really famous, like say Lionel Messi or something like that. And like if there are certain combinations found in nature that you have to take for granted, and those won't be found by our random selections.
Right. So again, I think Shaon also pointed this out. I'm not saying that teams are present only in cancer. Okay. And I'm not saying that teams in cancer are different than teams in development. They may be the same team. We don't know. We don't have data at that resolution to the same experiment being done in a non cancer cell line versus a cancer cell line, and you do all perturbations and see whether you arrive at the same network or not. We don't have that kind of data to make that claim one versus the other. Now, having said that, something which I did not talk about is the role of mutations, right? So when you have mutations, they may change the wiring dramatically depending on which master regulator is being mutated, and that may change the network such that you go from epithelial and mesenchymal dominated network to a hybrid dominated network, and that may be the difference between homeostasis versus cancer.
Okay, Mohit here. So I have a question related to reduction of this dimensionality of your team players. So I think it is more relevant in terms of drug resistance or how the drug might respond to targets, right? So can we map down to very few important team players so that we can even predict more drugs in the team? And also how we can switch from one to another state. So if we know the master regulators and how they are changing between states or teams, so in terms of targeting or drug resistance, how we can actually achieve that in terms of reducing the number of nodes or teams?
So for instance, in the EMT example, what we are saying now is that and these experiments I would love to discuss with you, see if they can be done, that give tamoxifen together with EMT inhibitors.
Right.
Because tamoxifen is pushing at least a percentage of cells to being more EMT. So is tamoxifen plus EMT inhibitors which is a rational therapy based on detailed validation, can that be a better therapy than just tamoxifen? Then we come to the next level of questions, again, questions which, for instance, Shaon and Yogesh think about is, do you give them together? Do you give them at intervals? Do you give a holiday in between? If they give in sequence, then which comes first? So all of those are open questions.
So if we have a wet lab, what kind of data we should provide you in order to actually get that nodes which are the most important or the teams which are most important for response or resistance? So how we should plan this experiment, suppose if somebody wish to do that?
So ideal information would be that we have the presence, because results vary, as you also talked about, so much based on the cell line and different drugs. So ideally, if we have all these interactions that I talked about in the same cell line, is there
Right.
information on that?
Right.
So we can search the literature and see which is the most well-studied cell line and build a network only from that cell line.
Right.
So far, the networks we have been building integrate multiple cell lines
Integrated, right.
together.
We
They
can
sort of
still combine certain cell lines which have a similar behavior in terms of one drug, or maybe have a similar phenotypic
right. So that
attitude.
level of similarity,
Right, has
that
to be there,
threshold has to be
because we don't want
carefully.
to map down to one cell, at least to epithelial versus mesenchymal, or
Yeah.
luminal versus basal,
Correct.
something like that.
So some categorization needs to
Some
be made,
basic
and yeah,
categorization.
so stricter than what we have right now.
Right. So
But
any data on sequencing, or I don't know, we can also build on metabolomic data, such kind
so
of nodes.
sequencing doesn't provide causative information in
Right.
most cases. So what information is more helpful to us is perturbation experiments.
Perturbation experiments.
And again, the more credible the perturbation experiments are, not just overexpression and downexpression, but binding site, mutation of the binding site. So our demands build up quickly in terms of when we
Sure,
want more
keep
credible
us
data.
updated.
Wondering if the hybrid states are somehow related to like drug persistent or drug tolerant states, because that is also a step somehow between, like, sensitive to resistant.
Absolutely. Anyone who wants to address that question is more than happy to come work with us. We are trying to address that question right now, and I hope that by the time we meet next, we'll have some answers. But that's our current hypothesis, that hybrid and or mesenchymal have some connections with persisters. But I think in breast cancer literature, persisters have not been studied as well, right? It's mostly lung cancer that was. So that's now, for that, we'll have to create a network of EMT in lung cancer and then see whether that pattern holds.
So I was curious to know whether this is done on single cell transcriptomics data and a perturbation study. Can similar kind of work be done for genomic data? For example, in a targeted therapy, EGFR mutations are occurring. In the starting of the treatment, if there is one mutation, like a sensitive mutation, that is L858R, which is sensitive to erlotinib, which is a TKI. But after a course of time of 6 months or so, they EGFR develops other mutations as well, which are resistant to erlotinib.
Right.
So if we have the data of post, or even a time span data of one month, can a study be designed in a such a way that we are able to understand if there is a specific pattern of mutations occurring? Because till now studies are there where everyone understands if there is a single mutation, they tell whether it is a resistant or resistant or sensitive. But there are very less studies where the combination of mutations is studied. So can we design a study in which we can understand the combination of mutations together and tell whether erlotinib can be suitable for that kind of condition or not?
Right. No, that's a great question. Right? So far, we have been talking about phenotypic data. Now, can we overlay or get information from genomic data on that? So one thought is that, at least I don't know the literature, that whether the presence of a genomic mutation can increase or decrease the likelihood of other mutations. In these networks, these are causative networks, right? A is changing the levels of B. Whether one mutation is changing the frequency of the other mutation or not, I am not sure the data exists, or at least I don't know of. So without that, building causative networks becomes a challenge. Then you can do associative studies, and club them in different behaviors and do some multivariate kind of analysis that is often being done. But I'm not sure about causative connections.